In this video, we're going to talk about enzymes, how they function, and then how we can regulate enzyme function. So here's an example of an enzyme catalyzed reaction. So sucrose, you eat table sugar, that's a disaccharide, a glucose and a fructose held together. To get into your blood and to be able to be used by your cells, it's got to be broken down into a monosaccharide. So we've got this enzyme sucrase that can bind to it, hold it in such a position that that bond can be broken with a lot less energy. You don't have to heat it up really high. You can break that energy very quickly. So enzymes speed up reactions and allow for reactions to occur at body temperature. So let's look at a little bit more about enzymes. And a good analogy, this was in your textbook freshman year, I don't know if you remember it, but we have reactants and they have more energy. They're up here in this high pond. We have products, they're down here in this lower pond. Again, these represent energy levels. But to get the energy out of those reactants, we got to get them up over this barrier. That's called activation energy. And it usually takes a lot of energy. Remember in the calorie lab, you had to heat that food up to 451 degrees before it would start to break down and release the energy. So enzymes are like lowering this big brick wall. They lower the activation energy so that your body can break down food at about 98.6 degrees, your body temperature. And that's a lot easier and more efficient for living organisms if we got these enzymes to help govern chemical reactions. So let's look at what this looks like in a little bit more sciencey view. So here's free energy we have over here. So we have high energy reactants, we have low energy products, and that change in energy, which we call delta G, the change in free energy is right there. But again, we've got to get up over this hump, and if we have enzymes, it lowers that activation energy so that the reactions can occur much more quickly and with much less activation energy or heat energy to get them going. So here's how an enzyme works. So here is what we call the active site of the enzyme, and it's going to bind to this molecule right here. It's called a substrate. The substrate is whatever molecule that enzyme binds with. Now it doesn't quite always fit perfectly. In fact, there's this idea of this induced fit, that sometimes the enzyme has to move a little bit, or sometimes we need a little enzyme helper in there to help bind it to the substrate to get them to kind of match perfectly. We used to call this a lock and key, like it was a perfect lock for one key. But we now know that the key actually, or the lock, has a little helper in there, and those are called coenzymes. So let's talk about some of those, or cofactors really. A cofactor is something that actually helps an enzyme in a reaction or govern a reaction. And the coenzymes are actually vitamins, and a vitamin is basically an organic molecule that serves as a cofactor. So vitamin A is a good example of one of these. And if you don't get enough vitamins, these proteins, these enzymes in your body can't function properly. Vitamin A is obviously associated with enzymatic action in your eyes, and you'll eventually lose your eye function and go blind if these enzymes don't get enough vitamin A. There are all other cofactors that are inorganic. They're minerals, so things like iron and calcium and magnesium. Those are needed by certain proteins, certain enzymes, again, to bind with substrates and help them to break down and govern these reactions. So cofactors could be organic molecules, which we commonly call vitamins, or inorganic, which we commonly call minerals. And a lot of those trace nutrients that we studied early in biochemistry are actually cofactors that are needed by enzymes for proper function. Environmental factors can also affect enzyme activity. We're going to do a lab where we look at a lot of these environmental factors in some detail, but here's two very obvious ones. So temperature, as temperature increases, reactions tend to increase because temperature is the measure of kinetic energy in something. So as molecules move faster, more substrates will bump into the active site, and that reaction will go faster. But you can see here it reaches sort of a optimal value, and then it goes down. And we know that enzymes and proteins, when they get too hot, they get denatured. All those secondary and tertiary bonds that give them their special shape start to break and reattach and form in different ways, and they become denatured. So you can see that for something in your body likes something about 37 degrees Celsius, that's about 98 degrees. If you're a thermophilic heat-loving bacteria, you have proteins that can deal with a lot hotter environment because you've evolved in these hot sulfur springs. So they have a different optimal temperature, but they see the same kind of increase and then decrease in as we get away from that optimal temperature. Okay. We also talked about pH. Now, pHs can denature proteins, again, because we are changing the number of hydroxide or hydrogen ions. That's going to interfere with those charged regions on the protein. So those, again, for that secondary and tertiary structure, we're going to break those bonds, and we're going to cause changes in them. So if you're pepsin is an enzyme in your stomach. It can only function at a pH of about 2. Okay, it breaks down polypeptides into dipeptides, into 2 amino acids. Then when those dipeptides go into your intestines, there's an enzyme called trypsin. Trypsin can only digest at about a pH of about 8. So what do we have to do? Our body has to adjust the pH of all the food contents from the stomach and raise it to a pH of 8 so that those proteins can be broken down completely into amino acids by trypsin. So this is a good example of how homeostasis is really important, right? We've got to keep the acid environment in the stomach, but then immediately adjust it in the duodenum for the intestines so that the pH can be at 8. But these are 2 factors that affect enzyme activity, and we're going to investigate these in a lab in a lot more detail. So now we need to talk about how we can regulate enzyme activity. And this is important because sometimes enzymes are overactive and we need to shut them down, or certain toxins can interfere with enzymes and bind them up, and then we can die, get very sick and die. So one thing that can happen is you can compete with the active site. So if we have something called a competitive inhibitor, that's a molecule that will bind to that active site and block the substrate from being able to bind to it so the enzyme can function. So this is basically competing with the substrate and blocking it. There are also non-competitive inhibitors. They can bind to some other site on the enzyme, like back here, and when they do that, they cause the enzyme to change shape. So now that active site no longer fits with that substrate, and it's no longer going to be able to break down that substrate. So an example of that is a very common blood pressure medicine. It's called an ACE inhibitor. And what ACE inhibitors do is, some people have high blood pressure for genetic reasons. They just produce too much of this angiotensin, this hormone that causes blood vessels to constrict and causes high blood pressure. Normally when you're stressed or when you're in a fight and flight response, you want your blood pressure to go up so that you can function at a high level. But again, some people produce too much of these hormones or constantly produce them, and they have high blood pressure, chronic high blood pressure. So what do? Well, you can take an ACE inhibitor, and there's a series of enzymes that activate other enzymes in this whole process. And one of them is this enzyme called ACE, and this will bind to this ACE enzyme and basically prevent it from activating angiotensin II, which is required to cause blood vessels to constrict. So by binding to that, this drug keeps people that normally have high blood pressure, can keep their blood pressure at a normal level. And unfortunately, they need to take it every day of their life to keep binding to those sites on the ACE protein so that they can maintain proper blood pressure. But it works amazingly well. So this is a way we can actually develop drugs to treat people who have certain abnormalities or overexpression of certain genes for certain proteins and enzymes. So here's another example of a way we can regulate enzymes. There are certain compounds that can bind to and keep proteins in their inactive form. So enzymes can be turned on and turned off, and if we have an inhibitor, it can keep them turned off. If we have an activator, that can bind to them and keep them in their active configuration. So certain drugs can be activators or inhibitors. So you can imagine you can turn on certain proteins that can then activate certain enzymes and keep them on, or when you want them shut off, you can produce more inhibitors, which can then shut off those genes, shut off those enzymes. I'm sorry. And again, here's an active and inactive form, and sometimes substrates in what's called cooperativity can make sure an enzyme stays turned on. So if there is a substrate that is being broken down, that will keep the enzyme in its active form. If that substrate is no longer present, then the enzyme can go into an inactive form. So like if you're consuming a lot of milk, you're going to need to produce the enzyme lactase. So that lactose will bind to and keep that lactase in its active form, for example. And when there's no longer milk in your diet, maybe that will turn the lactase into an inactive form, so it's not sort of there trying to digest milk all the time. So that's an example of how a substrate can actually bind to and keep an enzyme in its active form. Sometimes in metabolic pathways, we see that end products can actually bind to and turn off an enzyme. So for example, producing the amino acid isoleucine, there's a series of enzymes that are involved in that pathway. But once we start to produce too much isoleucine in the cell, we no longer need to keep producing it, right? We want to have negative feedback. In other words, when we produce too much, we want to stop it so that we don't end up with too much. So as isoleucine levels rise, isoleucine is going to bind to the enzyme, bind to what's called an allosteric inhibitor. So it's going to basically bind to another part of the enzyme and turn it off. And it's going to then deactivate the enzyme so that it will no longer initiate this pathway to produce more isoleucine. So this is again a negative feedback mechanism whereby our body can regulate how much of something we're producing. When isoleucine levels go down, we'll no longer bind allosterically to the enzyme, and it will activate that active site and initiate this process of producing more isoleucine. So a negative feedback in terms of regulating a production of a key amino acid. So these are some examples of enzymes and how they function, and that's it.